coming today. It's my honor to present the guest speaker today, who's going to be giving the Julia and Patricia Kingsbury Memorial Lectureship, which is a lectureship that's done yearly in the area of breast cancer. Dr. Max Wisha is our guest speaker today. He's the Madeline and Sidney Forbes Professor of Oncology, recently named the Forbes Professor of Oncology. I knew of the Forbes because of Dr. Wisha. I was fortunate enough and honored enough to be able to take care of Sid's sister when she died from metastatic cholangiocarcinoma as a result of my relationship with Max. He's also the founding director and emeritus at University of Michigan, having founded the Comprehensive Cancer Center at the University of Michigan, and is known in the NCI world as the longest surviving cancer center of all NCI designated cancer centers. I believe you were the cancer center there in excess of 20 years. You were the cancer center director? 27 years. Wow. Okay, that is quite impressive to put up with that for 27 years. But I think what Dr. Wisha is best known for, and why I began my collaborations with him years ago, is his work in cancer stem cells, specifically as they relate to breast cancer. I would say that he's probably the founder or one of the godfathers of breast cancer stem cells, and was probably one of the first, if not the first, to co-publish in this area, and has pretty much led the pack. And I was very fortunate to get to know him better when we were both awarded the Komen Promise Grant as co-PIs several years ago, looking at racial disparities in cancer stem cells that brought us all the way to Ghana and harvesting samples from the Ghanaian patients, as well as many of our own. And so he's been on many, many different committees throughout the years at national and international levels. I believe now is a standing member of the Scientific Advisory Board of the National Cancer Institute, and has recently stepped down as the Cancer Center Director at University of Michigan to be able to focus more of his energies in his laboratory, specifically, as I stated, working in the breast cancer stem cell arena. And with that, I believe he's going to be talking about breast cancer stem cells today.
Can everybody hear me okay? Is the mic on? Good. Well, I think I'll just talk from here, and it's really a pleasure to be here. And as Pat said, we've actually been friends for many years, so it's particularly then nice for me to be able to come. And what I'd like to do is to talk about cancer stem cells, but I really want to put it in a very large context of where do I think the most exciting areas in cancer research are, and how does this really fit together, and what are the big problems that we face in cancer therapeutics. Of course, near and dear to Pat's heart. But I'm going to do that from the point of view of talking about breast cancer, and specifically breast cancer stem cells. So this is just a summary chart of how well are we doing in fighting breast cancer. So this is just a chart of deaths in the United States and UK from breast cancer. And it's interesting that the death rate went up every year until 1990, and then it's come down rather steeply. And when this has actually been looked at, it's continued to come down, that probably about 30% of this is due to increased screening, detecting cancer earlier, and about 2/3 is due to our development of much more effective adjuvant therapies. And I'll say a little bit more about that later. Why have we been effective with adjuvant therapies? That's kind of the good news. But the bad news is, and by the way, so women is about 30% less likely to die of breast cancer today than in 1990. That's quite an improvement. But the bad news is, for women with metastatic breast cancer, the needle hasn't really moved very much. And a matter of fact, the life expectancy, even with all our new targeted therapeutics that everybody is, of course, so excited about, justifiably so, the average life expectancy of a woman once she develops metastatic disease has only changed about 6 months. And I would argue that for virtually all of the common malignancies of solid tumors, that's the case, that we may have done well in some early stages of disease, but in metastatic disease, we really haven't moved the needle very much. And why is that? And what I'm going to talk about today is kind of three areas, but of course I'm going to focus on the stem cell component. But what I really want to talk about is that the reason that we haven't done better is the singular problem that really perplexes us in cancer therapeutics, and that is tumor heterogeneity. That is the feeling or the realization that within any given tumor in any given patient, there is a tremendous heterogeneity. Part of this heterogeneity is genetic, and 95% of the research has been on this area, the genetic heterogeneity. And of course, we've come a long way in doing next-gen sequencing. In our cancer center, we sequence every patient who's going to go on clinical trials. But in fact, using targeted therapeutics, especially when they're being used the way they are now, one at a time, has only moved the needle very little. And the reason for that is this heterogeneity. But what I'm going to show you is that this is only the first level of heterogeneity. And superimposed on genetic heterogeneity, there is an epigenetic heterogeneity, which is a reflection of the differentiation state of all the cells within a tumor. And over the last decade or more now, work not only in our lab, but in many labs have shown that this differentiation hierarchy has tremendous implications therapeutically, because targeted therapies, as well as a more traditional chemotherapy and radiation therapy, have differential effects on cells depending on their differentiation state. And cells that are in the most primitive differentiation state, that we're going to associate with this term cancer stem cells, which I'll describe in more detail in a few minutes, that these cells have very different dependencies on cell pathways for their growth and survival than do the more differentiated cells, even with the same genetic makeup. So this is an epigenetic heterogeneity. And finally, at the end of my talk, I'm going to talk about what's the role of immunotherapy in targeting this heterogeneity. So let me start with, as I often do, is talking about what is the cancer stem cell hypothesis, because I think it's been kind of misunderstood, and people often ask me, so is this in favor, is it not in favor? I think those are kind of, in a way, foolish questions, because the real question is, what are we really saying, and what is the scientific evidence? But I think it also is important, because what I'm going to focus on today is what are the challenges in going into the clinic, and I think it's fair to say that the onus is on proving that targeting cells with these stem cell characteristics improves patient outcome. That's the bottom line, and I'm going to talk about the challenges of this today. So the cancer stem cell hypothesis really has two separable components, and both are actually very important, and both have been studied very heavily from a very basic scientific standpoint. The first is, where do cancers come from? This also has very big practical importance for cancer prevention strategies, because I think that the conventional wisdom until probably the last 8 or 10 years was that carcinogenesis is really completely hardwired into the genetic makeup of cells, and that one essentially had any cell which, if it acquired the proper combination of mutations, activation of oncogenes, inactivation of tumor suppressor genes, would become a malignant cell. What's become clear from preclinical models and now in clinical examples is that that's not the case. And as a matter of fact, the differentiation of a state of a cell at which time it acquires these mutations is actually critical. And the bottom line is that cancers, I think that, we can talk about this in the question and answer session, but I think that I can comfortably make the statement that cancers arise in cells that either have or acquire the capacity to self-renew. Self-renewal is a property that is unique to stem cells or to immune cells. Every other cell in your body, once it has the signal to proliferate, is not the same as self-renewal, because other cells other than stem cells, essentially, the differentiated progeny express a different gene program than the one generation earlier as they differentiate, whereas the stem cell can make at least one other stem cell or two, and in a sense then is immortal. This doesn't mean that all cancers come from tissue stem cells. They may, because these cells already self-renew, so they're good targets for carcinogenesis. But there's equally good evidence in many malignancies, including leukemias, and the best example, that progenitor cells, which normally can't self-renew, acquire a mutation that imparts self-renewal characteristics. And so these become cancer-like stem cells. But it's not equivalent of saying that a cancer stem cell has to come from a tissue stem cell. And I'm not going to focus on this today, although we certainly are doing, and others are doing research in this area. What I'm going to focus on is what about the heterogeneity of tumors once they form, and what are we talking about with talking about cancer stem cells? Because I think the evidence is that cancers are driven by these cells that have stem-like properties, that these cells have a lot to do with treatment resistance and metastasis, and that's what I'm going to really be talking about. So the system that we've worked on is the breast, and of course this is important because breast cancer is such a big clinical problem, but the basic principles of stem cells and cancer stem cell biology really are generally applicable. And a matter of fact, many of the pathways that we're targeting now in moving into the clinic are the same across many different disease types. So in the breast, one has a stem cell that is characterized by the ability to give rise to other stem cells, including those that are quiescent, and these then self-renew, particularly during early pregnancy and during puberty, and form these cells that are progenitors and then are cells that make milk during pregnancy. We know then that through these mutational events, one then generates the heterogeneity of a cancer stem-like cell. We used to think that different molecular states of breast cancer may actually come from cells at different levels of differentiation. But more recent evidence suggests that there may be common progenitor cells that are extremely prone to form breast cancers, and that the different molecular forms of breast cancer really don't result from different cells of origin, but from the mutation profile and different states of arrest of growth. So Pat mentioned that we actually started some of this research, and I want to show the original article that we published with Michael Clark that was really the first to look at stem cells in any solid tumor. But really our work was based on the pioneering work of John Dick in Toronto, where he showed in the 1990s in human leukemia that if you took human leukemias and tried to transplant them into immunosuppressed animals, only a small fraction of cells had the capacity to transform the leukemia. Now initially those cells resembled stem-like phenotypic markers on the cell surface with CD34 and CD38. More recently it's clear that there are other markers too, but we then sought to determine whether human solid tumors also had a hierarchical organization. And what we did then was to test protein markers on the surface of cells that would be able to enrich cells that had the ability to transplant the breast cancer when put into immunosuppressed non-SCID mice. And this just shows our initial paper then in 2003, in which 200 cells that expressed CD44 but not CD24 were able to form tumors, whereas 100-fold cells, more cells, 20,000 that didn't have this phenotype formed no tumor. And furthermore, we could serially passage cells, and only cells with this phenotype would transfer the tumor, and cells without the phenotype wouldn't. And moreover, when we examined this tumor, even though the tumor was initiated by CD44 plus 24 minus cells, the resulting tumor recapitulated the phenotypic heterogeneity of the initial tumor so that only 1 to 5% of the cells express this phenotype and 95 to 99% of the cells express this phenotype every time when we serially transferred. Now what I want to tell you is kind of where kind of the punchline of what the whole part of my talk is going to be is so what, and what does this really mean for the clinic? And what I'm going to tell you is kind of what some of the conclusions are, and then I'll show you some some of the examples that over time we've looked at, and then show you what does this actually mean and how are we going to translate this into more effective therapies for our patients. What we have come to realize is that if the cancer stem cell model is correct, or to the extent that it's correct, many of our models that we've used to develop new therapies or to assess the efficacy of these therapies may be faulty. The primary way that we've developed new therapies or assessed the efficacy of therapies is by examining the effect of these therapies on the size of tumors. So in preclinical studies, we measure tumors in mice. In the clinic, we do scans and we measure tumors, and any of you that are clinicians know that the so-called RECIST criteria, which everybody uses as kind of the gold standard of you measure a tumor, and if it shrinks by more than 50%, you say the compound is active. What that has resulted is us producing many treatments and compounds that can cause regression of tumors, but they don't affect the lifespan of patients. And as a matter of fact, for most tumors, including breast cancer, tumor regression has almost no correlation with patient survival. The two are actually only very, very loosely related. And I'm going to talk a little bit about why that may be, why these cells are resistant to therapy, and how are we going to target these cells. And then I'm going to talk about what kind of clinical models or clinical endpoints do we need to use then to even assess whether our therapies are targeting the cell populations that are really important or that we're looking at. And this is kind of the model, I think, that one can look at, that most of our therapies are effective at killing these differentiated cells, and so the tumor regresses, but it just grows back because the stem-like cells are still in existence. And this now shows that we're trying to design therapies to target cancer stem cells. An important question in this area is whether one actually can get more differentiated cells to de-differentiate and form stem-like cells, and I'll say a little bit more about that later because I think it's an important area, and our approaches in the clinic are to target both stem cells and their more differentiated progeny, not just one or the other, but the important part therapeutically is that the approaches and the targets and the pathways are different in these cells and these cells, and that's kind of the most important, I think, take-home message of this whole talk. Now, one of the first examples in the clinic for this model of cancer stem cells and being resistant to therapy came from our collaborator, Ginny Chang, who was down at Baylor, is now at Methodist Hospital. And we had shown in preclinical models that when you treated mice with xenografts, that if you treated them with chemotherapy or with radiation therapy, the residual cells were enriched in cancer stem cells. And what she then did was the first actual clinical study using the markers that we had defined for cancer stem cells to ask if this is actually true in actual patients. And this is a kind of design that we often use now in breast cancer called the neoadjuvant design, in which the woman has a breast tumor that's treated, so you can actually measure things before and after treatment, but you can measure the effect of the treatment on the cancer before you then remove any residual cancer. And what you found was that if you treated patients, as their tumors shrank down, if you measured the percent of stem cells, they actually went up after therapy, very much as in our preclinical models. Now, interestingly, an exception to that was in the 20% of patients who had HER2-amplified breast cancers. If the patients were treated with a HER2 blocking agent, in this case the oral agent Lapatinib, you actually got a shrinkage of tumor. Along with the tumor shrinking down, you actually got a reduction in the cancer stem cells. Now, interestingly, in this case, one had about a 23 or 4% of patients that had a complete pathologic response. We know that a complete pathologic response is associated with a good outcome. Interestingly, the complete pathologic response more than doubled with the HER2-targeted therapies. And I'm going to talk about that in a few minutes. Why is that? But when we first saw this, we immediately started to think that perhaps HER2 is so important in breast cancer because it's an important regulator of the breast cancer stem cell. Now, the other question is, why do stem cells increase in the presence of chemotherapy? And this then was reproduced in many other tumor types. There are dozens of publications in both some clinical models and in preclinical models that cancer stem cells are relatively resistant to a number of therapeutic agents. Matter of fact, we had interesting conversations this morning about DNA repair. DNA repair appears to be more efficient in tumor stem cells. But the other thing we found, which was somewhat surprising to us initially, is that in our preclinical models, if you treated mice with chemotherapy, not only did the percentage of stem cells increase, but the absolute number of stem cells actually increased. And that suggested that there was actually a stimulatory pathway that was being produced by the use of chemotherapy. And what we found then was this was actually due to the fact that when we treated cells with chemotherapy, it induced a damage response, and the dying cells secreted several cytokines, including interleukin-8, which turned out to be a very important driver of the cancer stem cell. We showed then here in a paper in Journal of Clinical Investigation, that the IL-8 worked on the cancer stem cell by binding to a specific receptor, CXCRI, which triggered self-renewal through activation of Wnt signaling. Now, interestingly, this is not only interesting because chemotherapy is actually stimulating the cancer stem cell, something that actually has gotten to me in trouble. I've had patients say, does this mean I shouldn't take chemotherapy? No, it doesn't mean that, but what it means is that that if we could block this pathway, perhaps we could increase the efficacy of chemotherapy. So an Italian company called Dompe had been making a small molecule inhibitor of CXCR1 that was quite specific, wasn't designed for cancer treatment. It was designed as an agent to permit transplant, organ transplant. And so we performed preclinical studies showing that if you treat tumors with a cytotoxic chemotherapy, docetaxel, you increase the cancer stem cells, but you could block that by blocking the CXCR1 receptor. So based on this, we went into then a clinical trial, and Ann Schott was one of our clinical investigators, performed a phase 1 clinical trial here with 33 patients. And this was patients with HER2-negative disease, and this was patients with paclitaxel with this stem cell inhibitor. I'm going to talk about a number of clinical trials targeting cancer stem cells, but one thing that's actually been interesting is that a number of these pathways, even though they may be shared between normal stem cells and cancer stem cells, have been quite non-toxic. After in a phase 1 trial, though, of course, it's really hard to know exactly whether this represents effects of chemotherapy or is this really better than you'd expect with this therapy alone. So Dompe has now initiated this last year a randomized clinical trial which will involve about 250 patients, half of which will be getting chemotherapy plus the stem cell inhibitor and half will be getting chemotherapy alone. And I think, of course, that's what we need in all of these kind of clinical trials. Now, I wanted to show you another example of why it's so important to understand cancer stem cell models in thinking about results of currently existing therapeutic strategies. Well, there was a lot of enthusiasm in the clinical community for the approach of using anti-angiogenic agents based on the pioneering work of Judah Folkman and others that tumors required a blood supply to grow beyond a certain size. And of course, a number of inhibitors have entered the clinic, including bevacizumab, which is an anti-VEGF antibody, or kinase inhibitors that also inhibit VEGF signaling. Now, interestingly, in breast cancer, and this has now been true in many cancers, this was a huge drug for about 5 to 10 years almost. It was a $5 billion drug, and Pat would know this better than I was. It was being used in all these tumor types. And the effect in the clinic was virtually the same in many tumor types, including breast cancer. So in breast cancer, when bevacizumab was added to chemotherapy, the patients had a better response, the tumor shrunk down more, and it appeared that they had a longer disease-free interval before the cancer progressed. And on the basis of that, the FDA actually approved bevacizumab for the treatment of breast cancer. However, when the patients were followed, what was found was that once the patients started to progress on bevacizumab, they had more aggressive disease. And the time that they actually lived was no different than if they hadn't gotten bevacizumab at all. So basically, it was without any utility. So they withdrew the approval. Now, women group, including some of my own patients that had been on some of these trials, protested. They said they benefited from it. But what was really going on? What did this mean? So this was an example where we looked at what was actually going on physiologically by looking at our preclinical models. And we had known that stem cells, normal stem cells, live in hypoxic environments. So even in your bone marrow or in the CNS niche, the oxygen tension around the stem cell area is actually very low. And so the issue was, are breast cancer stem cells regulated by hypoxia, and could angiogenic agents really be regulating that? This shows Sarah Conley in a study that she did then, which was to grow tumors, and you could see the tumors grow here. If you give the anti-angiogenic agent, you block the growth of the tumor. This is same in another model. These are the controls, and this is with the anti-angiogenic agent. You see the tumors are white. But what she found is if you look at markers of stem cell, and this was another marker that our group identified, which is a a series of enzymes called aldehyde dehydrogenases. I'm going to say more about this in a few minutes. And there's an assay called the Aldefluor assay, which allows us to very quickly look at the level of aldehyde dehydrogenases. And it's a rough, I think, indication of a stem cell phenotype. And you can see that the percent of Aldefluor positive cells is increased by the anti-angiogenic agent. But even more importantly, if you took tumors that were treated with this anti-angiogenic agent and reimplanted them into mice that were not treated, which is called a reimplantation assay, which is really the best assay for stemness, you can see that here with lower cell numbers at 5,000 cells, that the control cells, that the treated cells had a greatly increased number of tumor-initiating cells. And we went on to show that this was generated by hypoxia due to the anti-angiogenic agent. So this is a agent which is sensitive here to hypoxia. A hypoxia probe turns green in the presence of hypoxia. This is the control. This is sunitinib treated. These are green cells. And we counterstain this with the antibody to aldehyde dehydrogenase showing red. And you can see that we have increased stem cells right around the hypoxic zones over here. Now, why is this important? It's important because it's telling us again that if we have agents and we use them only on the basis of tumor regression, unless we consider the stemness of these other cells and figure out a way to target that, our therapeutic efficacy will be limited. So in order to figure out how we're going to get around that, we had to know the mechanism of how does hypoxia increase cancer stem cells. So what we found is that one of the characteristic or most important pathways that regulates hypoxia-related gene expression is the HIF pathway. And what we found here is that if you knock down here HIF-1 alpha, you can completely abrogate the effect of hypoxia on stem cells, but not HIF-2 alpha. So this is a HIF-1 alpha mediated process. And we went on to show that HIF-1 alpha then stimulates Notch signaling, which is an important developmental pathway, as well as Wnt signaling. And this was very important because it suggested a way that we might be able to combine agents that can inhibit cancer stem cells with anti-angiogenic agents. So many of you are familiar with the Notch pathway. There are 4 mammalian Notch receptors, 1 through 4. When Notch ligands like delta or jagged bind to the Notch receptors on adjacent cells, one has a cleavage in the membrane caused by an enzyme called gamma secretase, and the internal domains translocate to the nucleus where they activate a series of transcription factors. And the first Notch inhibitors utilized were then gamma secretase inhibitors, which could inhibit this cleavage process. And what we showed was that if you increase the cancer stem cells with hypoxia and you add in the Notch inhibitor, you could abrogate that effect. We could also abrogate the effect in an in vivo model. Here you could see the growth here, and then if you add the Notch inhibitor along with this. So based on this, we again initiated a phase 1 clinical trial along with Merck, and in this case we used the gamma secretase inhibitor along with the cytotoxic chemotherapy. Now, one of the limitations of all of these studies, though, is if we don't we can't use tumor regression as an endpoint. How do we really know that we're even hitting the stem cells? So one of the things we did in this study, along again with Jenny Chang, was to get serial biopsies before and after the treatment on this phase 1 study. You can see we treated 30 patients on this study, and these are the clinical results that we got. And what we did is we got serial biopsies and measured a series of tumor cell markers and showed that we could knock down the stem cells here. A number of randomized studies using other Notch inhibitors, including some from a company that I founded called Oncomed, are now in randomized clinical trials again. So we'll see whether adding these agents together really improves clinical outcome. But I wanted to also point out the problems here with clinical endpoints. Now, the story of HER2, I had mentioned before that HER2 is one of the most important targets that we use in breast cancer. And as clinical oncologists, if you ask what's the most important development that has changed the natural history of breast cancer, I would say that it's probably using HER2-targeted therapies in patients with HER2-amplified breast cancer. And one of the reasons that it's so important is that we've now shown that HER2 is a potent regulator of breast cancer stem cells. If you just take cell lines and transfect them with HER2 and measure the percent of ALDH positive cells, you can see how they increase here in every case when you when you overexpress HER2. And just as importantly, if you have a HER2 overexpressing cell line and you add Trastuzumab or Herceptin, you knock down the percent of stem cells. So we also went on to show, although I don't have time today, that HER2 is an important driver of cancer stem cells, not only in HER2 elevated breast cancers, but even in HER2 breast cancers that have only a single copy of HER2, where HER2 is selectively expressed only in the stem cells. So that's why it was missed. And we think that may actually account for the surprising finding that Herceptin may actually have utility when it's used in the adjuvant setting, even in women who don't have HER2 positive breast cancer. But when women become resistant to HER2 targeting agents, what accounts for that? Well, what we found, again, we modeled this in our preclinical model, and we took cells that overexpressed HER2, either that we transfected in or endogenously overexpressed HER2, and we knocked out PTEN. Now, PTEN, as you know, is a very important negative regulator because it regulates AKT signaling. And the most important genetic abnormality in patients that's associated with Trastuzumab resistance is inactivation of PTEN, often through a gene silencing, not through mutation. So we mimic that in our preclinical model. When we knock down PTEN, the cells become completely resistant to Trastuzumab. And what we showed was, and this is Hassan Korkaya showed, that these cells are resistant to Trastuzumab because they activate an independent growth factor pathway, and again involving a cytokine, in this case interleukin 6. So when you have a sensitive cell, what you have then is Trastuzumab inhibits this pathway, but when you block PTEN, you activate NF-kappa B signaling that activates IL-6 and setting up a positive inflammatory loop. And these cells are completely resistant to Trastuzumab, and in fact, these cells actually downregulate the cell surface HER2 expression. So even if one is using an immunoconjugate, an immunotoxin like TDM1, you can't kill these cells because the HER2 goes down. But they activate an IL-6 pathway. So just like we did with IL-8, we then thought, could we block this pathway and overcome Trastuzumab resistance in these cells? And indeed, what we found was we actually could do that, and we published this in Molecular Cell. So what you can see here is, first of all, if you just take you take a HER2 overexpressing cell line, and you can see that it's sensitive to Trastuzumab, but then the cells start to grow and they become resistant. If you block IL-6 receptor with an antibody called Tociluzumab, you can completely abrogate that. More importantly, if you take the HER2 overexpressing cells in which we knock down PTEN, they're intrinsically resistant to Trastuzumab. So these are the cells growing, and these are the PTEN knockdown cells. They're completely resistant to Trastuzumab, but they're completely sensitive to the IL-6 inhibition. Now, interestingly, this drug, Tociluzumab, is already approved by the FDA. It's a Roche drug, a Genentech drug that's approved to treat arthritis because interleukin 6 is involved in arthritis. So it's taken us now about 3 years to work with Genentech to convince them to design this trial. And now it's going through our IRB. Monica Burness, one of our young clinical investigators, will be the PI of this trial. We'll be enrolling patients that have Trastuzumab resistant HER2 positive breast cancer. We'll be looking at the serum levels of IL-6, which we can find are quite elevated in these patients, and we'll be treating them with the Tociluzumab. Now I'm going to get back in a couple of minutes of what is there about this combination of Trastuzumab and Tociluzumab that allows this to be so effective when they're used together. We'll actually see how effective it is because this is the clinical trial. And the clue to this was looking at the markers that we have defined as being able to enrich for cancer stem cells and starting to look at tumors to see what were these markers actually identifying within actual tumors. So our initial publication showed that cells that were CD44 plus 24 minus were able to initiate tumors in mice. A subsequent paper showed that ALDH expressing cells were able to induce tumors in mice, and some cells overlapped the two. But when we started staining tumors and looking at these markers, what we found was actually surprising. What we found was that they were actually at any one time identifying different cells within a tumor. So here you could see the edge of a tumor, and here we stained these for CD44 here in green and 24 here in purple, and ALDH1 here in pink. And you can see that the 44 plus 24 minus cells tend to be at the outside edge or the invasive edge of the tumor. In contrast, more centrally, we found the ALDH positive cells, and here's a cell that is one of the hybrids that's expressing both CD44 plus 24 minus and ALDH. So what's the difference between these cells? So this study was done by Su-Ling Liu, again in collaboration with our investigators down at Baylor. So what we did is we took a series of primary human breast tumors and by flow cytometry isolated the 44 plus 24 minus or the ALDH positive cells and then did molecular profiling. Initially when we did this we were using microarrays, now we use RNA-seq. And what we found was very interesting. There were some genes that were common to both of these stem cells, and those were genes that we thought were involved in the self-renewal process. But interestingly, there were another set of genes that were essentially completely oppositely expressed in these two stem cell populations. The 44 plus 24 minus cells expressed cells that we knew were involved in this state of transformation called EMT, or epithelial mesenchymal transition. Now, many of you know that there's been a lot of work looking at the role of EMT in metastasis. And as a matter of fact, Bob Weinberg from up the road at Harvard had published some seminal papers showing that there was a relationship between EMT and stemness of cells. And what I'm going to show you is that he was right, but only probably half right, because the 44 plus 24 minus cell indeed upregulates these EMT transcription factors and metalloproteinases, but the ALDH cell is depleted in these markers. In contrast, if one looks at the epithelial-like markers like E-cadherin and these claudins, that they are downregulated in the EMT-like cells, but upregulated in these ALDH positive cells. And you can see that if you look at a proliferation marker, Ki67, it's enriched in the ALDH cell, but depleted in the 4424 cell. And when you look at these cells, you see what you might expect here. So the ALDH positive cells are epithelial-like, whereas the 44 plus 24 minus cells are mesenchymal-like. And what we found is that these states are not hardwired into the cell, but in fact are plastic. And if you take either the 44 plus 24 minus cell or the ALDH cell and culture it in vitro, or if you put it in a mouse and grow a tumor, in either case, you get an equilibrium of these two cell types that is characteristic for that tumor. And this just shows that they come back to equilibrium no matter which cell you actually put into the tumor. And so this actually summarizes a lot of research that we've done subsequently to look at what regulates these switches. What we think now is that the cancer stem cell state is a very plastic state, particularly between the epithelial and the mesenchymal-like forms of the stem cell. And that the microenvironment of the tumor is what controls these switches back and forth between EMT and MET-like states. And within the microenvironment, with a variety of growth factors and things, which I'll just talk about in a minute, we have a series of microRNAs as well as long non-coding RNAs that regulate these switches back and forth. And interestingly, what we found is that the pathways that regulate these two types of stem cells are different. And remember that these are plastic. So the epithelial-like stem cell shown over here is highly driven by the HER2 pathway, and that's why Trastuzumab is very effective at killing these cells, and HER2-expressing cells are quite luminal and epithelial in their characteristics. When you lose PTEN, you shift these cells to the mesenchymal EMT-like state in which they downregulate HER2 but become sensitive to IL-6 signaling, and that's why Tociluzumab inhibits this state of the stem cell. So this model suggests that in order to effectively target stem cells, we may have to target both states of the stem cell. Now, why is this so important in metastasis? What we think happens is that the EMT occurs at the invasive edge of the tumor, controlled by the microenvironmental cells, by pathways such as TGF-beta and some inflammatory mediators, that through microRNAs make these cells into an EMT-like state that then circulate within an EMT-like state and form a micrometastasis. But the micrometastasis is dormant if it stays in an EMT-like state, unless it can flip back to an epithelial state. This matches very well what I actually see in patients, which is that circulating tumor cells are largely Ki-67 negative, and I'll talk more about the circulating cells in a minute. And that micrometastases are very much 44 plus 24 minus, but the ALDH turns on when these cells then become epithelial and grow. And the dormancy of tumors may relate to them sitting in an EMT-like state. So then to get a metastasis, one has to have a stem cell, otherwise you wouldn't have the proliferative capacity to form a macrometastasis from a micrometastasis, but dormancy may relate to these EMT-like cells. So this is a summary now of a number of pathways that we and others have identified as regulating the cancer stem cell. Some are cell type intrinsic, some are from the microenvironment, like these growth factors. And these now are drugs that are in clinical trials to target these stem cells, a number of which are being done at our institution. This is just a list of 9 clinical trials being done at our institution. But one of the biggest challenges we have is how are we going to monitor patients on these trials? And it opens up kind of a bigger discussion now of how do you monitor heterogeneity in real time in patients so that you know that your therapies are attacking the right cells in the right patient, both from a genetic as well as epigenetic standpoint. And as I mentioned to you already, the circulating tumor cell provides a potential liquid biopsy to do that, particularly with a model that the seeds that are responsible for metastases are in fact cancer stem cells. So we've worked now quite a bit with our engineering colleagues, and we've had some collaborations here with Pat to try to develop new technologies to interrogate cancer stem cells. But in order to really look at heterogeneity, we think you're going to have to use single-cell analysis to really interrogate these cells. So Sunitha Nagrath, one of our engineering colleagues, has designed microfluidic channels that can separate white blood cells and circulating tumor cells. The established assay that has been approved by the FDA, called Cell Search, utilizes the antibody EpCAM to capture circulating cells. But as I've shown you, and we now have a lot of data, that the majority of circulating cells have very low EpCAM or EpCAM negative because they're in the EMT-like state. So Cell Search is only the tip of the iceberg of what it's capturing, and that's why we felt we had to use label-independent methods to capture these cells. I don't know if you can see this, but this actually shows just fluorescent labeled tumor cells and white cells that are going on different channels with our microfluidic devices. We now are working with the Fluidigm company to perform single-cell analysis of these cells. The initial studies that I'm going to show you now involve using single-cell multiplex PCR so that we can simultaneously look at 96 genes in each individual cell. But where we're going now, and which we already have set up, is to use RNA-seq on single cells. So we'll actually be able to look at the complete gene expression profile on single cells. And this is part of the device. This one is commercially available yet. We're working with devices with them to develop new devices that aren't available yet. But this shows that we could take cells from the circulation and they go into these single cell devices, and then we can do multiplex PCR in these cells. Now, which genes are we going to look for? So we selected a set of 96 genes based on 2 things. Half of them were candidate genes, so we took things that we knew were in stem cells, like ALDH and CD44, CD24. The other half of the genes, though, were done not on a candidate approach, but an unbiased approach. So we took our profiling data of ALDH positive tumor cells and 44 plus tumor cells and did a next-gen sequencing and selected the 40 genes that best discriminated these cells. And based on that, we came up with these markers, including ones that you see here in blue that are associated with epithelial cancer cells, ones that are in red that are associated with mesenchymal cancer stem cells, as well as you'll see, markers of white blood cells to eliminate any of them that may have been captured in our devices. This is with the kind of data that we generate. So each of these is an individual cell. Each of these is 96 genes. So you basically get 10,000 data points worth of PCR from these experiments. And when we then took tumor xenografts, and this is a primary tumor xenograph of a triple negative breast cancer, putting them through our 96 gene profile, We then can analyze the 96 genes statistically by using principal component analysis with principal component 1 on this axis and principal component 2 here. And you can see that the epithelial-like stem cells separate completely from the mesenchymal-like stem cells in this array. And interestingly, if we put in white blood cells, they completely separate from the EMT and the MET stem cells. So we can now separate all of these cells. And now this is an example now of looking at patient circulating cells where we can see these different principal component analyses. And these are the kind of plots we get. So each of these is a different marker. And this shows the gene expression of each of these genes across a series of cells that are circulating in patients. And we can see both epithelial and mesenchymal cells, and then we can do principal component analysis, in this case 3D. We have PC1, 2, and 3. And here you can see epithelial-like circulating tumor cells, mesenchymal-like circulating tumor cells, and some mesenchymal-like cells, and some cells that aren't stem cells at all. Having done about 30 patients now, our general findings are that in the primary tumor, about 1 to 5% of the cells express stem cell markers. In the circulating cells, it's about 50% have stem cell markers, and a little more than half of those are mesenchymal. We see some that share mesenchymal and epithelial markers, which have some of both. And this is some work here that we're also doing along with Tony Koleski as well as Pat, using a technique called CyTOF technique. What this allows you to do is proteomics. So we would like to be able to interrogate single cells at the proteome level as well as at the RNA expression level. And what this technique allows you to do is to detect up to 40 proteins on each individual cell using antibodies. What's nice about this is you can look not only at proteins like stem cell markers, but you could also look at signal transduction pathways. And this is an example here. So this is from a patient. These are circulating tumor cells, and it's distinguishing them from all of the different kinds of white blood cells. And this will also allow us to see whether there are any immune cells that are involved in this. And this actually shows here looking at signal transduction pathways, phospho-ERK, phosphostat, phospho-MEK, so we'll be able to look at these on individual circulating cells. The other thing that we found, which is now being reported regularly in the literature though, is that not all of the cells that are circulating are single cells. And as a matter of fact, there's now substantial evidence that these clusters of circulating cells may actually be much more metastatic than the circulating single cells. Matter of fact, a paper just came out this week showing in a preclinical model they're about 100 times more effective at metastasis. So one of the postdocs in our lab, Ramdan, is engineered filters that can catch clusters and single cells by having a pore size that is different. And so on the upper filter, we capture clusters, and on the lower filter, we capture single cells. And this just shows what we can do with these. These are the filters with different sizes. We can do this kind of analysis. What we're interested in is what's actually in the cluster. What are these cells in the cluster? And this is interesting. What we're finding now is that the cluster is a complex microenvironment. There's more and more data that invasion and metastasis of cells involves a collective migration of a sheet of cells in the circulation. And that we think that what happens is that you have a cluster of cells that essentially are carrying their microenvironment with them when they form metastases. And what you can see in this cluster, which is quite interesting, is these are epithelial-like cells, and these are mesenchymal-like cells that are 4424 on the outside over here. And we think through this kind of analysis, we'll be able to look at these clusters. What we really want to do is to look at them in patients in real time when we're actually looking at patients. So why is this so important? Because I talked about heterogeneity in this talk, but I think we have to think about heterogeneity a little bit differently than the traditional way of just looking at the genetics. Tumors arise in self-renewing stem cells, but these stem cells can exist in an epithelial or a mesenchymal-like state, and these stem cells then differentiate into a whole hierarchy of differentiated cells. In addition, there's some evidence that some of these may de-differentiate, but again, as I said, that's controversial. But these stem cells themselves are not static, and the stem cell can mutate so that this stem cell is different than this stem cell. And it also forms colonies. So you have a degree of heterogeneity that's both genetic going down this way as well as epigenetic. And our challenge is that these may depend on different pathways. Now, how are we going to ever be able to solve the cancer mystery when we have so much heterogeneity? Well, one way may be combinations of inhibitors, and I know Pat and her colleagues are most interested in that. And we think we can select at least the best combination rationally by looking at single-cell genomics and proteomics and pick the best combinations of inhibitors. But there's one system that has evolved to deal with heterogeneity, and that system is the immune system, because we know that immune cells can attack many different invaders simultaneously and essentially evolved to actually do that. Now, one of the whole things in the cancer stem cell field is when we started a lot of these experiments, we and everyone else took human tumors and put them in immunosuppressed mice. And so, by definition, we were dealing with very abnormal immune systems. Once the excitement of using immune therapies in cancer has become a reality with checkpoint blockers, we now have gone back and started to look at that. And what we and others are now finding is that the cancer stem cells are highly regulated by the immune system, both positively and negatively. And so the key is, can we now use the immune system to attack cancer stem cells, because it can deal with the heterogeneity that might exist in these cell populations. So the first question is the stem cell and the non-stem cell recognized as being different? Well, what we did here was to actually look at that by trying to make specific vaccines that were generated with using dendritic cells to boost the immune response, but generating a vaccine using either a stem cell or a differentiated cell. But this time, instead of using immunosuppressed mice and putting in human cells, we have to go back to mouse models using syngeneic mice that have intact immune systems. And so we did this in a number of models, and I'll just show you one example now. So this is a 4T1, which is a well-known mouse mammary tumor model. It grows very quickly and forms lung metastases. Fortunately, the same markers that we use for human stem cells work just as well in these mouse stem cells. These are ALDH positive cells. These are ALDH negative cells at the same inoculation, and the ALDH positive cells are much more tumorigenic. So what we did then was to make a vaccine using ALDH positive cells or ALDH negative cells in this 4T1 model. Oh, I just want to show you that one of the, of course, immune blockers that prevents the immune system from recognizing a tumor is the PD-1, PD-L1 system, where the checkpoint blockers are involved. So we looked at PD-L1 expression on ALDH positive cells and negative cells, and we consistently find about a twofold enrichment of PD-L1 on the ALDH positive cells. So it's even more immunosuppressive. The good news is, because we have checkpoint blockers, we can also look at using PD-L1 blockers along with ways of attacking the cancer stem cell. So along with Chao Li and Fred Chang at our cancer center, we developed the following model in the 4T1. What we do is we put in the 4T1 cells, we prepare a dendritic vaccine that's either stem cells or bulk cells. But now in one group we also add anti-mouse PD-L1, and we compare the effects on metastasis in the lung. And I'll only just show you one experiment because of time, but we've done this with a number of tumor models. So these are the number of lung metastases. And you can see here that using a cancer stem cell vaccine, we can reduce this. I don't show it actually on this slide, but using bulk cells, we get almost no activity. And this is anti-PD-L1, and this is the combination. And you can see the combination knocks out these metastases. So what I've shown you is that there may be hope of attacking the heterogeneity of tumors, both by understanding the molecular profiles of these tumors at single cell resolution, or secondly, by using the immune system, or potentially both, to again attack this heterogeneity. So I think we're at the stage now where there are about 100 clinical trials around the world now going on in the early stages of targeting cancer stem cells. Using immunotherapies to target cancer stem cells is still in the developmental stage, but I suspect we'll see clinical trials with this over the next year or two. And I think that will be, again, an ultimate test of whether targeting this kind of heterogeneity really improves patient outcome. So thank you very much.